All right, let's just get started so we have enough time to get to the presentation and questions. Hello, everyone. Welcome. My name is Caroline Morin. I'm a scientific analyst at the Gilbert Family Foundation, and it's my pleasure to welcome you all to this new presentation in the GFF NF1 seminar series with our speaker today, Dr. Jeffrey S. Chamberlain. Dr. Chamberlain is a professor in the Department of Neurology, Medicine, and Biochemistry at the University of Washington. And he has been studying muscular dystrophy and approaches to therapeutics for the past 30 years. The focus of his research has been on the mechanism leading to muscular dystrophies, the function of dystrophin and dystrophin-associated proteins, and genetic approaches to therapy. His group has performed extensive structure function studies on dystrophin and how dystrophin-glycoprotein complex is assembled in striated muscles, and has pioneered methods for gene delivery to skeletal and cardiac muscles. As part of a longstanding collaboration with the lab of Stephen Hauschka, they have also pioneered the development and the use of muscle regulatory cassettes to drive gene therapy expression in muscle while minimizing immunological and toxic effects of ectopic gene delivery. Beyond his many accomplishments within the DMD world, Dr. Chamberlain is also an advisory board member for the Gene Therapy Initiative at GFF. And we are really excited to hear his talk today, focusing on the lessons learned through his development of gene therapies for muscular dystrophy and how we can apply that to neurofibromatosis type 1. So Dr. Chamberlain, if you want to take it away.
Okay. Thank you, Caroline, for that nice introduction. So just by way of introduction, maybe to partly repeat some of the things we just heard, I don't actively do research on neurofibromatosis. I'm learning a lot about the field and have followed it for many years and getting increasingly updated. My work is really more focused on the muscular dystrophies, but hopefully by sharing some of the things that we've gone through over the last 25 years or so, maybe there'll be some things that are useful to the NF1 community. We'll see. I'm mainly going to focus on the dystrophy work, but I'll try to highlight differences and similarities and issues that are going to be important for neurofibromatosis as well. So with that qualification, let's get started here. Okay, so just a quick overview of what has been happening in the field of Duchenne muscular dystrophy. So there are actually many different types of muscular dystrophy. The number keeps growing despite the human genome being sequenced decades ago or whatever it was. There are more than 50 different kinds. Duchenne muscular dystrophy is thought to account for about half of all cases, and that's the main form that we work on. The couple of similarities and differences between Duchenne and NF1 are outlined here. It's probably a pretty simple overview, but the dystrophin gene, which is defective in Duchenne, is the largest known gene. It's more than 2 million base pairs in size, so that's about 100 times bigger than a typical gene. The NF1 gene is also enormous. It's about 350 kb. I'm not quite sure where that ranks in the order of the largest genes, but I'm sure it's in the top 10, if not the top 3 or 4. The inheritance is slightly different. Duchenne is an X-linked disease, so it's recessively inherited. NF1 is more or less autosomal dominant. It's on an autosome. Mainly, it's considered dominant because of its role as a tumor suppressor gene, but in other ways, it can function more recessive in its inheritance. The genes themselves are quite distributed. Dystrophin has 79 major exons, 8 of which are alternatively spliced, but there are also 6 additional exons 1s, reflecting the fact that there are actually 7 different promoters in the dystrophin gene, and these different promoters are expressed in different tissues, and they express, in many cases, different isoforms of the dystrophin protein. NF1 has a slightly fewer exons, 62, with 4 of which are alternatively spliced. And the frequency is both is quite high. They're among the most common human genetic disorders. Duchenne affects 1 in 10,000 newborns. It only strikes males, so it's 1 in 5,000 males. but 1/3 of all cases arise from a new genetic mutation. So it's a new mutation syndrome, and the dystrophin gene is thought to be the most has the most new mutations of any known human gene. So it's an issue in terms of diagnostics and treatment strategies, because even if you were to cure every single patient, new ones would continually arise. Some similarities arise with NF1. NF1, I believe, is thought to be the most common human genetic disorder, a single gene disorder that is, with about 5% of cases arising from a new mutation. So some of the history of this disease, the dystrophin gene was cloned back in 1985. And is generally thought to be the first gene that was positionally cloned. And some people argue that that was the beginning of the Human Genome Project, because it was the first time a gene had been cloned without any knowledge of the protein that was encoded by that gene. So it was isolated based on its position on a chromosome and various tricks that Lou Kunkel at Harvard applied to bring out a gene that turned out to be frequently deleted in many patients. And that was a key to his cloning strategy, which I'm not going to go into here, although it was kind of interesting. NF1 was cloned not much longer. I believe it was only the 3rd gene to be positionally cloned. And it was isolated by Francis Collins, who was at the University of Michigan at the time, where I was also. And a little piece of trivia, Francis Collins and I used to work pretty closely together. And he and I actually submitted the first funded gene therapy program project grant that was picked up by NIH, and it was focused on a couple of different diseases at that time. So what progress has been made over the years on Duchenne muscular dystrophy? As I mentioned, you know, a lot of kinds of dystrophy, but dystrophin being the first gene identified and being the most common dystrophy has received the vast majority of the attention in these disorders. However, dystrophin actually interacts with many other genes, some of which, or excuse me, many other proteins, a number of which are modified by even more proteins and genes. And by when the dystrophin gene was cloned, that led directly to identifying many of these other genes. And it turns out that a lot of the genes that encode proteins that interact with dystrophin, when they're defective, they cause different types of muscular dystrophy. So there's been at least 30 different genes that have been cloned based on their protein interactions with dystrophin or the dystrophin-glycoprotein complex. And most of these cause different types of muscular dystrophy when they're defective. A bit of a timeline here. Mouse models, I think, are critical for developing therapies for genetic diseases. The main one that's been used in the muscular dystrophy field is the MDX mouse, which was originally described in 1983 by Graham Bowfield in England. The first muscle-specific gene regulatory element was identified in the muscle creatine kinase gene back in 1985, a work that I did together with Steve Hauschka. The dystrophin gene itself, as I mentioned, was cloned in 1985 by Lou Kunkel. And an important observation soon after that by Kay Davies' lab at Oxford was that there was a patient found that had a 1 million base pair deletion. And you know, if you recall, the dystrophin gene is over 2 million base pairs in size. So this deletion took out half of the protein coding region of the dystrophin gene, yet this individual was very mildly affected. And in fact, he died in his late 70s, according to Kay Davies, and never lost the ability to walk. So that was a remarkable observation from a disease that at that time was uniformly fatal before the patients reached the age of 20. So that, you know, to have somebody missing half the gene walking into their late 70s was extraordinary. And that really was a key observation that helped spur some of the early work in gene therapy in this disorder. The dystrophin cDNA is about 14 kb in size. My lab actually assembled a lot of smaller cDNAs that had been isolated by several labs. We put together the first full-length dystrophin cDNAs from both the mouse and the human gene in the 1989, 2000 region, and we started using those to make transgenic animals. And one of the first ones we made was a fairly simple design. We simply re-expressed the full dystrophin protein in the MDX mouse model for Duchenne muscular dystrophy and showed that we could eliminate all symptoms of the disorder. And that was back in 1993. Now the issue there at the time was that, generating a transgenic mouse is a reasonably simple technology in the laboratory, but it's not something you would ever conceive of doing in humans. So the goal then turned to the idea of, well, now that we know that gene replacement can cure Duchenne muscular dystrophy, is there a way to actually replace a gene in patients? And it's quite a challenge, particularly with muscle. It's probably more of a challenge with neurofibromatosis because in the muscular dystrophies, almost 40% of the human body is muscle mass. So that's an enormous target area to try to deliver new genes to. And again, with NF1 is going to have a lot of different cell types, tissue types that will need to be treated, that obviously goes well beyond simple muscle tissue. In the 90s, a lot of the work in this disease was turning towards identifying vectors. In other words, gene delivery vehicles that could be used to transfer genes. In our case, we're interested in muscle, with many other groups working on this for different tissues. Liver was probably the greatest attention, a lot of interest in cancer cells and things like that. And many different vector technologies were tested. Some worked better than others, but none of them were ideal. And the limitation of most vector systems throughout the years in terms of trying to introduce new genes into the body has been issues with the immune response. The human immune system is pretty good at identifying foreign invaders, and unfortunately, all the early efforts at gene therapy were the reaction of the recipients, being animal models or even some patients in the early trials, was generally to launch a very robust immune response and reject the treatment as a foreign invader. This started to change in the late 90s with various groups that had been working on vectors derived from adeno-associated viruses. And I'll come back to AAV technology in a little bit here. But the initial studies involved intramuscular injection. So just targeting a very small region of one muscle in the entire body. And those early studies led by Barry Byrne and Jude Samolsky and a couple of others showed that AAV, unlike really any other vector system that had been tested to that point, was stably expressed. I think the longest studies that were published early on went out about a year and a half and showed that they could get stable expression of genes in muscle at the site of injection. So that led to a lot of proposals that, oh, well, now we can easily cure Duchenne muscular dystrophy by injecting a patient about 10,000 different times to inject the gene here and there and all that. And people, I think, fairly soon came to the realization that that was a ridiculous proposition that would never work. So then, people started thinking about, well, how can we possibly deliver a gene body-wide? And a variety of approaches were tested to try to do that. And I think, modestly speaking, a key breakthrough came from my own laboratory, where we took a couple of different vector systems and tried directly introducing them into the bloodstream through intravascular injection. And when we first started doing this, everything simply went to the liver. We couldn't get anything to muscle. But I had a couple of persistent postdocs that continued with this, and they said, well, let's just increase the doses massively and see if we can get anything to start leaking out of the bloodstream and into muscles. And in fact, with AAV, but not with any other vector system we tried, when we got up to what at the time we thought were just astronomical doses, they actually still are astronomical doses, we could actually see a very sudden and dramatic increase in the amount of vector that was leaking out of capillaries and getting into the underlying muscle tissue. So that really set the stage for us. We now knew that we had a vector system that could be delivered body-wide. But one of the questions was, well, what do you deliver? Because we've got a 2 million base pair gene and a beautiful vector system that only holds about 5,000 bases of DNA. So that was an issue. But fortunately, for many years, while trying to figure out delivery systems, we had been doing a lot of structure function studies of dystrophin, starting with the full-length clone, moving on to some of the deletions that were found in mild Becker patients. And we actually identified a number of very small versions of dystrophin that had surprisingly high levels of functional activity. And I'll come back to that in a little bit here, but we were able to show that some of these, but not a lot of them, were able to have a dramatic effect in ameliorating the phenotype after a body-wide delivery using these AAV vectors. And many years later, that led to the first systemic microdystrophin gene therapy trial, which was led by Jerry Mendel at the Nationwide Children's Hospital in Columbus. And since then, human trials of that technology have really scaled up, but they have not yet been approved for routine prescription use. So just to back up now and go through some of those steps and some of the things that that we worked on over the years. The goal really for gene therapy of Duchenne muscular dystrophy, and I have Becker muscular dystrophy up here also. Becker is just a name for a milder version of Duchenne muscular dystrophy. It's actually the same disease, but I'm not going to go into that much here. It's not that important. So you need to find ways to replace or repair the dystrophin gene. At least that's the strategy that I've been pursuing for 30 years, literally in my lab. In the early days, the most people I talked to about gene therapy said to me, basically, gene therapy will never work. You're wasting your time. And most of the field was focused on developing small molecules or drugs to try to combat dystrophy. And unfortunately, none of those drug delivery strategies has really worked to any significant extent. Gene therapy is now finally working, but it was a very long haul to get to this point, and it required overcoming a lot of skepticism, not only in the general community, but in the patient community, and unfortunately in the funding community. It was not unusual to submit grant applications in the late 90s proposing gene therapy strategies to have the reviewers come back saying this will never work. You're an idiot. So I think it's changed a little bit. So we'll see how that goes. Anyway, the real needs, as I've indicated earlier, are body-wide systemic gene delivery. In our case, the target tissue is muscle, and that requires some sort of vector. As I alluded to a minute ago, AAV vectors have significant potential, and to this day are the only vector system that is able to affect systemic or body-wide gene delivery. And again, the first study on that was run by a postdoc in my lab, Paul Grigorovich. The other challenge is the size of the gene, and this is a challenge with neurofibromatosis as well. We're dealing with the largest known gene. To my knowledge, the dystrophin gene has never really been isolated in a single clone. There's a yak clone that's been going around for many years that had the entire gene, but they found it had a small deletion in it, just like in patients. But through years of work, the 2.2 million base pair dystrophin gene has been whittled down to what we call microdystrophin, which there's a lot of different types of microdystrophins there, but they're on average, they're about 3.8 kb. So that's a tremendous miniaturization. The other issue that's turned out to be important to us, and I don't know how this is going to relate to issues with neurofibromatosis because of the diverse tissues that need to be targeted, but we felt it was important to restrict gene expression to muscle cells. The main target, obviously, of gene therapy for muscular dystrophy is muscle, but most vector systems, and including AAV, depending on how you work with it, can even with the systemic delivery methods, they can go body-wide, not just to muscle tissue. So the way to restrict gene expression to muscle, we decided would have to come from the promoters, the regulatory cassettes, the promoter enhancer sequences that were in the vectors that would drive expression of your transgene, in this case, the dystrophin or microdystrophin. And again, fortuitously, when I was in graduate school, my project was to identify regulatory elements from a muscle-specific gene, and that was the muscle-creatine kinase gene. I was a graduate student with Steve Hauschka, and through that work, again, it led to the isolation of the first muscle-specific enhancer element. And Hauschka's lab has since spent the last 35 years developing a variety of different regulatory cassettes from the muscle-creatine kinase gene. So this has led to a number of therapeutic advances. Gene replacement is now in clinical trials. I'll come back to that at the end. Another approach is gene editing, which I'm not going to go into too much today, but I know that's an important potential therapeutic for NF1. Gene editing is coming along really well in the Duchenne field. It's not quite there yet, but I think it has tremendous future potential. Okay, so let me go through some of these steps in a little more detail. So the key to a lot of this, again, is adeno-associated viral vectors. So these AAV vectors have been around for many years. It's a naturally, AAV itself is a naturally occurring human virus, but it has the peculiar feature of having never been linked to any type of human disease or even any kind of human pathology. So it's perhaps a kind of an evolutionarily unusual virus and it's not able to replicate on its own. It requires helper viruses. And that's where the unfortunately quite confusing name of this virus has come from. It's called adeno-associated virus. And the reason for that is that AAV was originally identified as a contaminant in an adenovirus experiment. So it's, again, AAV is not able to replicate on its own. It needs proteins from other viruses, and adenovirus is the one that people typically use to grow AAV, but herpes virus and a couple of other viruses can also be used as helper viruses to grow AAV. The reason I say the name is unfortunate is that there is no end to the number of people that confuse adenovirus with adeno-associated virus. They are completely different viruses. There is really no relationship whatsoever other than the fact that adenovirus can be used as a helper to grow AAV. But the real reason it's kind of unfortunate is that adenovirus was an early candidate for gene therapy vectors, but it led to disastrous results, and its first use in the clinic led to the death of a patient known as Jesse Gelsinger in a ornithine transcarbamylase gene therapy trial. And that really set the field back many years. So we try to avoid the use of the word adenovirus these days, but it's inevitable when you're dealing with basically what is an adenovirus associated virus. Anyway, this is an interesting vector system when it's vectorized. In other words, you remove the viral genes and you use it as a gene transfer vehicle. It does not integrate into the human chromosome, at least under most conditions. So it persists in cells as an episome, and that has advantages and disadvantages. There are now millions of different serotypes of AAV. There's a couple of hundred naturally occurring ones, but many laboratories, and including some of the ones working on NF, of course, are engineering libraries of synthetic adenovirus serotypes. And like I said, there have been millions of versions of that are being tested in different labs. Many, but not all of them, are highly tropic for muscle. So we zeroed in on ones that were indeed muscle tropic. In general, the vector system has shown minimal immunogenicity. In other words, eliciting an immune response against the vector. Now, when I refer to that, I'm really talking about cellular immune responses, T-cell immune responses, things of that nature. All vector systems, when introduced into the body, do elicit a humoral immune response. So you get neutralizing antibodies activated. You also get other antibodies produced that are not necessarily neutralizing, and those are starting to appear to be quite important. And I'll talk about that a little bit at the very end of my talk. This vector system has a scalable production. There's a lot of ways being done to do that, to scale it up, to be able to treat thousands of patients as opposed to treating 5 mice in your own laboratory. And as I mentioned, it can be used for systemic gene delivery to many different tissues. And in our case, we focus on striated muscles. So what are the disadvantages? There's a number of disadvantages. It has a very small cloning capacity. It only holds about 5 kb. And again, for large genes, that can be an issue. The NF1 gene is 350 kb. So one has to think about what can you deliver that would fit into this size capacity. AAV vectors in general have a relatively poor transduction of quiescent stem cells in the body as opposed to more mature tissues. And they're not retained by dividing cells, so that it may or may not be important depending on your target. Again, with AAV as an episome, just like a plasmid, when you transfect a plasmid into a cell line in culture and they start dividing, you lose the plasmid. It's not retained. It's not part of the chromosome. Another disadvantage is that most AAV vectors naturally get into the liver. And part of that is that you don't necessarily need specific receptors to take up AAV vectors. Liver cells are highly fenestrated and can take up small vectors like AAV without needing a receptor-mediated event, although there are ways around that. And a significant issue is that many patients have been exposed to AAV natural viruses in their lifetime and have some pre-existing immunity, and that can exclude them from clinical trials. But that's a complicated area because there's so many different naturally occurring versions of AAV. Not everybody has a pre-existing immunity to all types. Generally, someone might have immunity to a couple of AAV serotypes, but not others. But there are a lot of open questions about how much cross-reactivity you might get, and that's going to be an important issue to expand this technology beyond those patients that have no pre-existing immunity. And at the bottom is sort of a generic structure of an AAV vector. As I mentioned, the difference between a vector and a virus is you no longer have any viral genes. It cannot replicate on its own. So the AAV vectors are produced in tissue culture, usually in human 293 cells, although there's a lot of other systems to make AAV. In general, the only thing from the virus itself are the two ends, the so-called ITRs, which is an inverted terminal repeat. You need the ITRs for replicating and packaging these plasmids into an infectious particle, the AAV vector. And generally, all of the viral genes are stripped out of this piece of DNA and replaced with a promoter element, a transgene, and a polyadenylation signal. And then you can package this up into an infectious particle by either co-infecting your cells or co-transducing your cells with either a helper virus or a plasmid that contains the relevant helper genes needed for replication and packaging of the vector. So here's an example of one of the early studies we did with AAV vectors, and this illustrates really two things. One, the difficulty in finding ways for systemic gene delivery, and two, what we refer to as a threshold effect for transduction. So here's an example. Again, in our case, I'm not sure you can see my pointer, but on the left, we looked at three different muscles: the heart muscle, the diaphragm, and the tibialis anterior muscle, and delivered a gene that encodes an alkaline phosphatase reporter gene. And this was done by intravascular infusion. And as I mentioned earlier, when we first started doing these experiments, we might inject, 10 to the 6th, 10 to the 8th, the viral particles intravascularly, and we wouldn't see anything. It would look just like these controls here. There was absolutely no alkaline phosphatase activity produced. It was only when we got up to, again, what I refer to as an enormous dose of in the range of 2 times 10 to the 13th vector genomes per kilogram body weight of the recipient animal, we started to see a little bit of gene transfer, particularly in the heart. And at least in the mouse models, most AAV vector systems, although not all of them, seem to have a preference for heart over other types of muscle. Now, what I'm not mentioning is liver. Again, you can actually get vector into liver at significantly lower doses, but we don't see any transfer to solid organs until we get up to much higher doses. And again, even then, we could see some gene transfer into the heart, but not into the skeletal muscles. But as we increase the dose, if you notice here, this is really only a 2.5 fold increase in dose. And again, 0 to 10 to the 13th gave us nothing. And now 2.5 times more gives us an enormous increase in transduction. We're starting to see some in different skeletal muscles, and you can keep increasing that up to the point where you start getting really useful gene transfer throughout the body. So a couple of important points to keep in mind here, though, are one, we see significant differences in gene transfer to different muscles. There are differences between cardiac muscle and skeletal muscle. There are also differences between individual skeletal muscles, and even within individual skeletal muscles, we see differences from muscle cell to muscle cell, which somewhat reflect both the anatomy of individual muscles, but also the muscle fiber type. So even a single muscle typically has a lot of different types of muscle cells in them. the simplest way to divide that is fast and slow twitch muscles, but there's also a lot of subdivisions of fast and slow twitch muscles. So we not only see a lot of variation from muscle and fiber type, but if you go beyond muscle, which we haven't had to, but you will in the NF1 field, I think you're typically are going to see differences from one cell type to another, depending on what organs you're looking at. The other thing, though, to keep in mind is that this is all highly dependent on which serotype of AAV you're working on. So this work I'm showing you here was done with AAV6. We see some differences with other serotypes of AAV. Some serotypes of AAV don't get in the muscle very well at all. So, every new serotype needs to kind of be individually tested. And importantly, there are so many new types of AAVs coming out that hopefully ones will be found that will be, better matches for each individual disease or cell types that need to be targeted. This is a graph of what I showed you earlier, just kind of making a point in some ways. On the left is a logarithmic scale of gene expression levels. And on the bottom here is a linear increase in dose. So this just makes the point that once you hit this threshold level where you start to get any gene transfer into your target muscle tissues, simple linear increases in dose then can lead to an exponential increase in the amount of gene transfer you see. But the rate of this transfer at different doses varies from one muscle to another. And I think that's going to be true for most cell types and serotypes of AAV, whether they're natural or synthetic, is that you're likely to see different transduction profiles, dose responses, depending on which vector type you're using. So let me talk a little bit about the dystrophin protein itself, which again is defective in Duchenne and Becker muscular dystrophy. So dystrophin is a cytoskeletal protein. It lines the interface of the muscle plasma membrane, which is more or less referred to as the sarcolemma. And it has a number of protein-protein interaction domains. So the dystrophin protein is pretty big. It's certainly not the largest protein, unlike the gene itself, but it's about 430 kilodaltons in size, and it interacts with a lot of other proteins. It links to F-actin filaments immediately below the sarcolemma. It actually interacts with gamma-actin, not with the contractile alpha-actin. There's many other protein interaction domains, and one of the key roles of dystrophin is to nucleate the assembly of this large complex of integral and peripheral membrane proteins together, known as the dystrophin-glycoprotein complex. And some of these proteins span the membrane and link to the extracellular matrix via laminin. And together, this dystrophin complex is thought to play a critical role in helping to dissipate the forces of contraction from within muscle cells out to the stronger connective tissue. So it participates in the lateral transmission of force. But this complex also assembles several signaling proteins that play some known roles, but a lot of, fortunately, still unknown roles in muscle cells. So, I mentioned earlier, a lot of structure function studies have gone on for many years in my lab and many others, of course. And part of the value of that, other than, trying to really develop a better understanding of what does dystrophin do, but it allowed the design of mini genes, if you will, by zeroing in on critical functional domains and being able to eliminate the less functional domains. Now, one of the earlier, in fact, the first earliest clue to this was this patient that I mentioned at the beginning that had a 1 million base pair deletion. The K. Davies identified many years ago. So this patient had a deletion that took out half of the gene. And the parts of the protein that were encoded on that deletion are indicated by this line here. So there was a deletion basically from this part of the protein all the way out to actually this part of the protein. This isn't drawn very well, sorry about that. And so we knew right away that this entire central part of the protein was dispensable for the vast majority of the functions of dystrophin. We've spent a lot of years really trying to refine those deletions, improve upon them in several ways. One of the things I should mention is that there's some problems with the naturally occurring deletions because one of the reasons the dystrophin gene is so large is that it has enormous introns in between the protein coding exons. And so almost all deletions that have been found in the dystrophin gene have breakpoints within introns. So they remove entire exons of the gene. And one of the problems with that is that the individual subdomains of the dystrophin protein are not cleanly encoded on individual exons. So in other words, you can have exon deletions that still allow for a protein to be made, but the resulting protein has partial functional domains that can interfere with its function. And it turns out if you just sort of completely ignore the exon intron structure of the dystrophin gene and design miniaturized proteins based on the known domains of individual functional domains, you can come up with proteins that are more functional than anyone's naturally occurring in the mild patients. And so that's something we adopted a number of years ago. And this has, you know, those of you that, if any of you follow what's going on in the Duchenne field, there's a couple of approaches to therapy that are sort of dependent on tricking muscle cells into skipping mutant exons. And that's potentially a very effective therapy, but it does have this disadvantage of making proteins that have partial functional domains that can interfere with the stability and the overall half-life and functionality of those modified proteins. And that's something with viral vectors, you can completely avoid that. The other point I wanted to make is that, you know, this deletion, again, was found in a very mildly affected patient, which is referred to as a Becker muscular dystrophy. One of the key differences between Duchenne and Becker is that most of the Duchenne mutations, which in fact are the vast majority of mutations that occur in this gene, prevent the production of any protein at all. So they're frame shifting deletions, they're deletions that remove promoters, or they're deletions that remove critical functional domains, like this cysteine-rich domain here is absolutely indispensable for function. If you have deletions that remove things of that nature, you get you don't get a protein made, or you get a very non-functional protein made, and you get the severe Duchenne muscular dystrophy. On the other hand, deletions that remove exons that have the right number of codons such that you don't disrupt the natural open reading frame of the messenger RNA, then you get tend to get the Becker muscular dystrophy. And the reason for that is that you'll end up with a protein that's being made up to the site of a deletion, and then the protein coding continues after the deletion because you have not disrupted the open reading frame. And when that happens, you make a protein that has the parts of the protein before and after the mutation. And in many cases, but not all, that leads to a milder phenotype because you've got a partially functional protein. And again, that leads to Becker muscular dystrophy. So here's an example of some of the studies we were doing back in the 90s, starting with the full-length dystrophin protein that we assembled many years ago. We started initially somewhat randomly making deletions in these proteins and putting them into transgenic animals. So again, this is not a strategy that's relevant to human therapeutics, but it taught us a lot about the dystrophin protein, what parts were most important and which parts can be removed without impact on the function. I'll just highlight a couple of key observations. One was this protein here we put together. We removed the entire so-called C-terminal domain of dystrophin that has a number of protein-protein interaction domains in there, and we were surprised to see removal of that domain had absolutely no impact on the function of dystrophin. This protein here was based on that very mildly affected Becker patient I talked about earlier that lived into his 70s. If we looked at force generating capacity of these constructs, which is something we often focused on, again, the wild type protein develops 100% normal force or strength, if you will. This C-terminal one also did, but the Becker patient only developed about 90% normal strength. And we thought that was a pretty good match to what we were seeing in the clinic with that individual. But again, if we actually made a larger deletion to remove one of these partial functional domains I was referring to earlier, we had now a synthetic protein that actually restored 100% strength. So that was a key to ignore these exon intron boundaries and go to the functional domains. We moved on from that just somewhat randomly, somewhat by design, removing additional domains. Here was an interesting construct when we removed the entire central rod domain of dystrophin, all of these so-called spectrin-like repeats that designated by Rs. When we took that out, that protein was non-functional. In fact, mice with this protein had a more severe dystrophy than the ones with no dystrophin at all. So that was an interesting observation. But eventually from this, we developed a lot of very small proteins that we decided to name microdystrophin. And the reason that I picked the name microdystrophin was pretty simple. Everybody had been referring to this construct from this Becker patient as a mini dystrophin. And the ones we came up with were really significantly smaller than that. And so to distinguish the two classes of protein, we started using the word microdystrophin. I should point out again, if you have any interest in the Duchenne field, not everybody uses the word microdystrophin. And there are other groups that have developed proteins this small that still call them mini dystrophins. And that's led to some confusion in the literature on what's the difference between a mini and a microdystrophin. And the answer is it's the only difference is who's doing the naming. So in the meantime, again, this is just a different way of representing these different proteins. This is, again, the protein that came from that original mild Becker patient. This was one of the early mini dystrophins, if you will. And then these are three different microdystrophins that we came up with. This one here, this so-called hinge 2 microdystrophin, was the very first one we designed. I actually put this into a grant application back in 1997, and it entered clinical trials in 2018 by Jerry Mendel. So that took almost 20 years to go from conceptualization to the first human clinical trial. This is the microdystrophin that's currently being used by Sarepta Therapeutics. It's also being used by Genethon in France, and Roche in Europe has said that they're going to start a human clinical trial with this protein. Now that's great. They're making a lot of progress on it, but again, this was the first one we designed. We've since come up with much better ones, and in fact, we've identified some significant disadvantages to this early one. So I kind of wish that some of these companies would keep up with current technology. One of the things we did was there's a so-called hinge domain. There's actually 5 or 6 hinge domains in dystrophin. Our earliest microdystrophin tried to keep as much of the normal structure as possible. So we really only had a single deletion in here, and then we took out this carboxy terminal domain. So this seemed to be the simplest way to make a microdystrophin, but this hinge 2 region, when it's brought up against this part of the protein, causes some interference and problems with protein-protein interactions. So when we switched from using this hinge 2 to the so-called hinge 3, all of those problems went away, and this protein here is more functional than the first one. In the meantime, we've also come up with newer ones, taking advantage of observations of other important protein-protein interaction domains. In particular, Dongsheng Duan's lab at the University of Missouri showed that this part of the protein was critical, or not necessarily critical, but really important for additional function of protein. And so we've inserted that into some of these microdystrophins, and this construct here is our current best in terms of overall functionality, although we have a whole new generation coming along where we hope to perhaps make them even better than what we have now. So we'll see how that goes. So again, this construct is being used by 3 different companies. This latest one is being used by Solid Biosciences, as well as Ultragenics, and we hope to come up with a newer generation shortly. So back to the idea now, combining these two things of taking a microdystrophin, putting it into an AAV vector, and infusing it into the vasculature of mouse models of muscular dystrophy. This is what we saw early on. This is just a wild type mouse, different cryosections from a variety of different muscles throughout the animal, stained for dystrophin. So this is a wild type mouse. This is a dystrophic mouse here. You actually see about 1 in 500 muscle fibers with some background dystrophin staining. These are so-called revertant fibers, which are thought to occur through some kind of back mutation event, which is to this day still has not been identified. But we include some of these regions in our photos for contrast. It's helpful. Again, they're only about 1 in 500 myofibers. This is from a dystrophic mouse treated with an AAV microdystrophin vector through an intravascular infusion. This is about 1 year after the infusion of the AAV vector. And hopefully you can see almost all the muscle cells in the body are now making this microdystrophin. This is a low power image of the gastric medius muscle showing essentially 100% dystrophin positivity. And again, this is about a year after the infusion. We've gone out almost 2.5 years and we don't see any loss of expression over that period of time. That's about as long as you can go out in the mouse. Others, I know Dongcheng Duan and Zhao Zhao have done some dog studies. They've gone out pretty far. I think Dongcheng has dogs out 5 years. Zhao Zhao had one dog that went out 7 years. It appeared to have fairly stable expression of microdystrophin. So it's reasonably long lasting, but we don't know how long it will last. I think it's unlikely to last forever, although there are others in the field that would disagree with that. Here's an example. Oh, this is the long time point. So again, this is 27 months after infusion into a dystrophic mouse with a microdystrophin. Wild type along the top are microdystrophin treated animals in the middle, and the dystrophic mouse on the bottom. I focused here on the diaphragm muscle and the heart. Those are the two critical targets in Duchenne muscular dystrophy. Normally, both of these organs show a lot of pathology at 2 years of age, and it's completely eliminated in the microdystrophin treated animals. Scaling up to see, can this go beyond the mouse models? Most of the work in my own lab has really focused on the mouse models. We've collaborated with a number of groups to look at the dog model, and those are Dongcheng Duan, as I mentioned, but also Joe Cornegie's lab, who was at Texas A&M at the time. And some of these studies have more recently been done in collaboration with Solid Biosciences. And this is an example of two things, really. One, at the very beginning of my talk, I talked about this threshold effect and changes you see with the dose escalation. We see that in the dog also, and it's undoubtedly being seen in patients as well. At what I call a low dose, but most people would probably call an enormous dose, 1 × 10⁻13 viral particles per kilogram body weight. We see only a small amount of dystrophin being made. A 10-fold increase brings that up significantly. Another 2-fold increase. We're getting to about 70% transduction of muscles in the dog. So not quite as good as the mouse model, but not too bad. And this is now the dose that most groups are actually using in clinical trials. So it's not a saturating dose. It'd be nice to go a little higher, but this seems to be about as high as you can go with safety standards with current vector systems being used. And this, it's a little hard to see here, but restoring microdystrophin also brings back many of the other, well, really all the other members of the dystrophin-glycoprotein complex. There's another way to look at this. This is more Western blot data, trying to quantify how much dystrophin is being made. It's a little difficult because we're dealing with a microdystrophin, which is a protein only 1/3 the size of the full-length protein. So Western blots can be quite deceptive in terms of, smaller proteins transfer better on Western blots and things like that. But again, at a 10⁻13 dose, even though we could see dystrophin being made by immunostaining of tissues, we couldn't see anything on a Western blot. But a simple 10-fold increase in dose changes that dramatically. Importantly, I haven't really talked too much about the muscle-specific promoters. In this case, this vector, this is, by the way, the vector that Solid Biosciences is using in the clinic. It has a muscle-restricted promoter, and so we don't see any gene expression outside of striated muscle tissues. And importantly, in the dog model, Joe Cornegie's lab has developed some very sensitive assays to measure strength of hind limbs in the dog. And with these microdystrophin infusions, he could show a very dramatic increase in strength in the dog model of Duchenne muscular dystrophy. And this was really the first time that anyone had been able to see a improvement in strength in this large animal model for Duchenne muscular dystrophy. So turning last part of my talk, I'm going to talk about the regulatory cassettes. What promoter enhancer elements do you want to use in your gene therapy vector? Now, for good or for bad, that kind of needs to be tailored to each individual disease. Early on, most labs interested in gene therapy were using viral promoter elements. Usually the immediate early enhancer and promoter element from the human cytomegalovirus, or so often referred to as simply the CMV promoter. one of the reasons for that is that's one of the strongest known promoter elements or gene regulatory elements. One disadvantage of that is that it is active in almost every cell type in the body. And that can lead, that maybe that's okay for a lot of diseases. In some cases, it's not okay. And reasons for that are that sometimes expression of your gene product in inappropriate tissues can lead to toxicity in those tissues. The other problem you can run into is that a promoter that's active everywhere in the body is often active in immune effector cells. And by delivering transgenes, particularly for a genetic disease, into immune cells, such as dendritic cells in particular, you can activate an immune response against that transgene, a particular cellular immune response, which is a destructive cytotoxic immune response that can lead to elimination of all of the tissues that you thought you just cured. So promoter use is critically important in gene therapy, and unfortunately, I can tell you what we've learned about muscle, but it's something that really is going to need to be worked out for every individual disease. So what we focused on again is developing what Steve Hauschka refers to as muscle-specific expression cassettes. Other people might just call those promoters, but it goes beyond a promoter because it contains enhancer elements as well as some other elements that can be critical for the levels of gene expression that you get. Most of Steve Hauschka's MSECs come from the muscle creatine kinase gene. MCK actually turns out to be the most abundant messenger RNA in muscle, but even though it's not the most abundant protein in muscle. But it was also just fortuitously the gene that led to the identification of the first enhancer element that worked in muscle. And again, that came out of Steve Hauschka's lab back in the 80s. So it's been a while. Some of the advantages, Steve has now developed several hundred different regulatory cassettes from the muscle creatine kinase gene and a few other genes. The advantages of these is they allow you to develop tunable expression for different muscle types. Initially, Steve was focusing on versions that would be as strong as possible in every possible muscle, but that's not always advantageous. Some muscular dystrophies require low amounts of protein. It can be toxic at higher levels. Some need to be expressed in skeletal muscles, but not in cardiac muscle. And in some cases, you might want really high expression in some muscle types, but not in others. And so it's if you go beyond the one or two muscular dystrophies and start thinking of all of them, you really need a wide range of different proteins. Early on, we showed that in this case with adenovirus, we delivered foreign proteins like E. coli beta-galactosidase. If we delivered them with the CMV promoter, we got a very potent cellular immune response against beta-gal. But when we delivered it with the muscle creatine kinase promoter, we saw no evidence of a cellular immune response against dystrophin. I'm not going to go into this in much detail. And it goes beyond just seeing an immune response. Some of our early studies where we were trying to develop systemic delivery methods, the easiest reporter gene we had was a CMV beta-gal reporter gene. When we delivered that body-wide at high doses to mice, all the mice died. They developed a potent immune response that destroyed the transduced cells of the heart. But when we switched to MCK cassettes, even with E. coli beta-gal, we saw no toxicity and no death from those studies. So yeah, this just briefly, Steve has developed hundreds of these things now that show an enormous range of activity compared to the wild type muscle creatine kinase gene. And they can be adapted for, I won't go into this in detail, but to give you gene expression in all the muscle fiber types in the body, depending on what your needs are. And an advantage of this is, one of the early reports, Jerry Mendel's first human clinical trial in Duchenne muscular dystrophy, he reported that it did not work because he induced a cellular immune response against dystrophin. And he was using the CMV promoter. So again, we've now carefully looked at this. This is in the dog model of muscular dystrophy, which does develop a very potent immune response against dystrophin under certain circumstances. But when we use muscle creatine kinase promoters, particularly the CK8 promoter, we see no evidence of an immune response against dystrophin in these dystrophic dogs. And this can have advantages for other systems. It looks like the slide's not very clear, but when we do gene editing, attempts at gene editing with the CKA promoter, we get good gene editing in muscle. Well, maybe not good, but reasonable gene editing in muscle, but none in liver. So it's a way to also prevent Cas enzymes from working in non-muscle tissues. So I'll just skip over the summary. I think I've covered that pretty well. So again, with where are we at now? We've got a vector system that gives stable expression for years in different animal models, significant improvements in muscle morphology and strength with no evidence of toxicity of a cellular or a cellular immune response, at least in the animal models. And there's a number of companies that have now taken this into the clinic. It's not a perfect technology. There's still problems we're trying to overcome. AAV vectors at high dose have been shown to elicit both thrombocytopenia and complement activation. So that's limiting the doses that can be used. That's something that maybe can be overcome with improved vectors. There's some hints that this may be caused by binding of antibodies to the AAV capsid itself. We'll see how that goes. Maybe improved vectors will get around that. More than 40 patients have been treated so far to date. There have been some serious adverse events. They've all been transient. All the patients are now doing well. We're seeing reasonable levels of dystrophin, but it varies from trial to trial and vector to vector. Let me just close out here with some of the things about perhaps applying this to other diseases. And Duchenne safety is really the biggest concern. I think we really need improved vectors to increase doses. This is going to require synthetic serotypes. It'd be nice to get a little more full reporting. Unfortunately, all the clinical trials are being done by for-profit companies, and so they don't necessarily tell you the real story of what's going on. I don't know what the best promoter is going to be for neurofibromatosis, not only for targeting, but to avoid immune responses. What are the best functional outcome assays? With Duchenne muscular dystrophy, turned out to be pretty slow in terms of the improvement that you see, and sure be nice to have better biomarkers or surrogate endpoints. Biodistribution is difficult. In all the Duchenne trials, people have only been able to take a single biopsy from a single muscle. It doesn't give you a good idea of what's happening body-wide. And we don't know how long this is going to last. Some of my friends in this field say AAV will last forever. I don't believe that. I think you're going to need to redose at some point, but that may depend on the tissue. How long it's going to last, we don't know. It'll probably last around 10 years, maybe 20. But you're going to have to redose at some point, and that's going to require improvements in immune suppression or improved vectors that don't elicit immune responses. And the problem, obviously, is either you may have pre-existing immunity, you may not, but once you get the vector, you've been vaccinated. So I will stop there. Sorry if I went a little long. Thanks.
No worries. Thank you so much, Dr. Chamberlain. This has been such an incredibly helpful view of the whole process of DMD gene therapy development. We'll vote one more minute left. If anybody has to jump off and has a question, please add it into the chat and we can definitely get that answer back to you. But for those of you who are around for a few more minutes to have your question answered live, please raise your hand if you have one, or also post it in the chat if you'd like me to read it. Okay, so we do have a question from Herb Sarnoff. How would you compare the gene therapy hurdles and challenges in DMD to those in NF1?
Well, it's difficult. Some of the similarities they're both enormous genes. There's no vector system that will deliver the intact genes. So we've got to go with cDNAs and probably truncated cDNAs to do that. Both the approaches require a lot of work in understanding the basic biology of these genes and their role in different tissues. One thing that may or may not be a little simpler with Duchenne muscular dystrophy is that our target has been muscle tissue. So, even though there's a lot of different kinds of muscle, it is still muscle. With NF1, it's not going to be so simple. There's a lot of different cell types that need to be targeted. Is that going to require just a generic targeting method that'll hit every cell in the body? And if so, are there going to be issues of any kind of ectopic expression induced toxicity or immune responses? What are going to be the ways around that? Promoter use can be important in limiting immune responses. you probably can't have a strict tissue specific promoter with NF1 gene therapy. So it may require alternate approaches, but maybe it won't be an issue though. Sometimes some of these diseases, Duchenne is a null allele disease. So generally patients make little, if any, of the missing protein. And NF1, that's not always the case. You can have some protein there that can help tolerize. But in cases where you have deletions or frame shifting mutations, that's may or may not be helpful. So there are going to be some certain challenges there. I think the vector systems, the AAV serotypes that have worked well for Duchenne may not be optimal for NF1, and so I think it's important for new vector systems to be developed. So again, I think we can learn a little bit from Duchenne. It's already made into the clinic, but there's always going to be very unique issues for individual disorders.
Great, thank you so much. We have 2 questions currently with people who have their hands raised. So why don't we start with Charlie Kurzbach?
Yeah, thanks, Jeff, for a great presentation as always. I learn more every time I listen to you. I had 2 questions, and if I only have time for one, we can you can choose, I guess. But one was, you mentioned that you think that redosing will be necessary. I guess my question is, for what reason do you think vector or expression will be lost and require redosing? And is that unique to Duchenne or generally applicable? And then I was also just wondering if you wanted to comment at all. I mean, you mentioned about AAV generally considered to be an episomal vector. Do you think that the low level of integration of AAV vector is going to play any role in any of the gene therapies being developed, or do you think that that's not relevant?
Well, those are both great questions. In terms of redosing, I mean, I guess a lot of it is a theoretical concern. Here's and it depends a little bit on the dose. If you can completely saturate all of the muscles in the body with microdystrophin, that should greatly stabilize those muscle cells. But at the doses we're currently seeing in the clinic, people are only they're not getting 100% targeting of muscle cells. So those muscle cells that are not targeted are still going to be dystrophic and turnover. So you're going to get a gradual loss of muscle. But would you have permanent retention of those cells that have taken up microdystrophin? Even there, I'm a little skeptical, and part of the basis for that is there's really 2 things. One, muscle is used for exercise, and it's clear every time you go out and do some exercise, climb stairs, go jogging, things like that, you do a little bit of damage to your muscles, and that's why your serum creatine kinase levels shoot up after exercise. And that damage is associated with partial loss of integrity of muscle cells and stem cell activated regeneration. So that's going to slowly dilute out an episomal vector. And there's other studies coming out from a couple of labs like Brad Olin suggesting that there is some natural turnover of myofibers, the nuclei in myofibers, that is, and replacing them with non-muscle nuclei. And if that really holds up in other systems, that's also going to lead to a dilution of the vector. So, it's just hard for me to believe that muscle, which is a tissue that does undergo some natural turnover during life, is going to retain an episomal vector for the lifespan of a patient. But hopefully I'm wrong, but that's my worry. I forgot your second question already.
What about the integration of the AAV?
Oh yeah, integration. It's a little hard to say if integration is going to be a problem. So about 1 in 10,000 vectors actually do integrate generally into naturally occurring double-stranded breaks. It's been a worry in other tissues because, with blood-borne gene therapies, like for severe combined immunodeficiency syndrome, integration of the vector near other genes has led to upregulation of those genes. In some cases, they're oncogenes, and that's led to leukemia in some of those patients. So integration of AAV vectors can be a concern for not only insertional mutagenesis, but for induction of cancer. Now, we tend to think that's not going to be an issue with the use of muscle creatine kinase promoters, because these are gene regulatory elements that are only active in non-dividing post-mitotic cells. So they're not likely to be able to activate other genes and induce uncontrolled cell growth. So, that's another reason I can get into to use tissue-restricted promoters. How that's going to relate to NF1 I don't know. In general, I would say that's not one of my biggest worries. I'll just leave it at that.
Thanks, Jeff.
Sorry, I thought I was on mute. Larry, if you want to go next.
Yeah, thanks. So thank you for that talk. It was really terrific, well organized and informative. Just one quick question regarding in the NF1 gene or a cDNA. Given the size of it, obviously what has, one thing that has to be done is to start understanding the absolute requirement for different domains to try to follow the mini gene strategy, microgene strategy. But another question is, if that turns out to be, if that doesn't turn out to be fruitful, the question of providing complementary domains that might somehow together form a useful protein. Whether you have any thoughts on that?
Yeah, I mean, if that will work for neurofibromin, that would be great. I mean, if you've got situations where part of the protein is made and a second domain can come in and they can dimerize or somehow otherwise work in parallel, that would be great. I don't know enough about the biology of neurofibromin to say whether that's feasible or not. There was a lot of early work in the Duchenne field on that exact same strategy, because the initial reports of dystrophin when it came out suggested it was a dimer, and so people thought, oh well, it's a dimer, we can put in half the protein with one vector, half it with the other. Well, later we realized it wasn't actually a dimer, and that doesn't work at all. So those kinds of strategies have not worked for dystrophin. Whether they work for neurofibromin, I don't know. It would be great. And again, developing the mini gene technology may or may not work. It's going to depend on, may depend on different tissues too, and the role of the protein. It's an important strategy to pursue. The other approaches, of course, are gene editing, which can maybe overcome some of that, restore normal expression of a protein, or perhaps even bring in missing domains. So those are going to be equally important strategies to take, I think.
Thanks a lot. I have to go to another call.
Great, thank you, Larry. And Stephen, go ahead. Thank you for your
Thanks.
patience.
You said the liver was a primary source of where AAVs were either activating or being taken up. And then when I look at your heart versus diaphragm versus tibialis anterior, you have greatest uptake in the heart, then in the diaphragm, and then the tibialis anterior. And that suggests to me that uptake is proportional to blood flow. And so where I'm going with this is I'm a microvascular physiologist. I study the microcirculation in skeletal muscle. And for it to get into the muscle fiber, it first has to cross the endothelium. And so I'm wondering now about being more effective in delivery if one considers the microcirculation, and more specifically where it's actually leaving the vascular compartment to enter the muscle. So precapillary arterioles are going to be tighter than capillaries, are going to be tighter than venules. And if it were up to me, I would try and make the vasculature permeant. In other words, break down the endothelial barrier at the site that I wanted to deliver the drug. And when I have spoken with people who study adenovirus strategies or systemic delivery, they don't really think about the microcirculation as something that needs to be addressed in delivering the particle to the myofiber, where from my perspective, it's essential. And so I'm offering the resources of my lab as somebody to collaborate with in this area, but I'm also curious what your thoughts are in terms of paying more attention to the vasculature when trying to deliver things systemically.
Well, those are really great points. I would love to talk to you more about that later. But I will say, interestingly, that is it's an area that received a lot of early focus in our field. In particular, a lot of that was set in motion by, I don't know if you know Hansel Stedman, who's at the University of Pennsylvania. Hansel published some early studies in the late 1990s trying to improve vector transduction by vascular permeabilization or dilation, things like this. And he was actually able to, so back to the larger virus, which is adenovirus, which again mostly goes to the liver. Hansel was able to show he could get reasonable distribution of adenovirus to limb muscles by co-delivery in the presence of papaverin and heparin and things of that nature. And we looked into that for a while with AAV. And interestingly, I mean, that's part of the way we got into some of these high doses and finding we could do systemic delivery was some of our early studies. We mixed in VEGF, or vascular endothelial growth factor, which has some vascular properties as well. And at some of the lower doses with AAV, if we added in VEGF or papaverin or some other compounds, we could significantly increase the amount of gene transfer into muscle, which we assumed was working, as you mentioned, so somewhere in the capillaries. Maybe I think Hansel has suggested it's a lot of the vector leaks out in post capillary venules and in that area. We haven't followed that up much. One of the problems we ran into, though, or maybe not a problem, was that we still were not getting uniform distribution of the vector throughout the body. So we started going up to higher doses of the AAV vector. And when we got to the doses that started giving us body-wide gene transfer, we no longer saw an effect of these vascular permeabilizing agents. And what we've concluded, although we didn't pursue this in the detail I would have liked to, is that some AAV capsids by themselves seem to have vascular permeabilizing activity. And so the idea was that once we went up to a high enough vector dose, these cofactors we were adding to enhance leak of the vector were no longer needed. And in fact, we did some studies where we showed we could just simply take AAV capsids with no vector genome in them at all and spike those into a vector preparation and see a significant increase in gene transfer. So again, the AAV capsids, at least some of them, have vascular permeabilizing activity. We haven't quite figured out why that is, but nonetheless, I think your point is a good one. If you can induce leakage out of the bloodstream into underlying tissues in a safe way, that could greatly lead to a reduction in the dose you needed, which would be tremendously beneficial. So, people have played around with a little bit. Most of the labs that were doing that have sort of given up. But the problem, I think, is most of the labs that were doing that work were not vascular specialists. So if that's an area you're interested in, it could be very fruitful.
If you're up for it, I'll contact you and we can talk about this more.
Okay, sure. Yeah.
Thank you.
All right, thank you so much. We do have one last question in the chat, Jeff, if you have a little bit of time, but if not,
Sure.
we can, okay. The last question that we'll do today is, how concerned or worried are you for the immunogenicity for either the microdystrophin protein or Cas9 protein for gene editing? And is there anything, particularly immunosuppressors, that might help overcome this?
Well, I guess I'm not enormously concerned. I mean, it appears that at least the muscle restricted promoters block immune responses against microdystrophin with what we've seen so far. Again, most of that is from animal studies. There's not an obvious immune response against microdystrophin in patients, but then again, the companies that are doing these studies are not necessarily sharing all their data, but nobody has reported a cellular immune response against microdystrophin, and there's been 40 to 50 patients treated so far. We'll see what happens, but I think it's looking pretty good so far. It may vary a little bit with the mutation. So there's 5 to 10 patients out in the world that have a complete deletion of the dystrophin gene. Whether they're going to be more immunogenic, I don't know. But for the majority of patients, I'm not too concerned about it. With Cas9, bacterial enzymes of that nature, I think we're gonna have to be a little more careful. Muscle promoters may well solve those problems too, but when you need to start delivering those in a more ubiquitous manner, we may need to bring in some additional strategies to prevent immune responses. I guess all I can say is that I know that that's an active area of study of many labs, many immunology labs that are really good at that work. It's something that we need to pay attention to, but I think it is a solvable problem. So, but I don't think you can just jump into the clinic and deliver high doses of Cas9 quite as easily as you might be able to other transgenes. So it's something that's going to have to be looked at carefully.
Great. Well, thank you so much for staying a little bit longer and answering all of these questions. And thank you so much for your wonderful talk, and to everyone else who participated. We're really excited about this seminar series. And if anyone has any other suggestions for future speakers, additional questions or comments, please feel free to reach out to me. And thank you again, Jeff, so much for your presentation.
My pleasure. Thanks.
All right. Have a wonderful day, everybody. Thank you.